Good morning everyone, I'm Candy. Recently, my mom has bought lots of cosmetic products back home after she has read the advertisement in a magazine. And I found that most of them emphasized coenzyme Q10 is included as an ingredient. So this makes me wonder the functions of coenzyme Q10 functions in cosmetic field. Nowadays, keeping beauty is not just limited to girls, but also boys. So, hope that all of you may get some useful information from my presentation today. Now, I would like to give you an overview of the introduction of my research topic, how coenzyme Q10 functions in cosmetic field. First, I will talk about the present situation, background information of skin and aging. It is followed by the introduction of coenzyme Q10, the gap of previous research, and lastly, the purpose and the structure of the project. Do you agree with the following sentence? Beauty is human nature. It is widely accepted that skincare and fitness of the body are two major concerns for women. And it is believed that skincare is considered as the lifelong business for women. So it is believed that cosmetic products are necessities for them. Nowadays, it is so prevalent that there is an introduction of coenzyme Q10 in cosmetic products, such as hand cream and face masks. Now let me give you a brief introduction of skin. Skin is considered as the largest organ, occupying 2 meters square area in human body, and it has various functions. For instance, it acts as the barrier to prevent the entry of bacteria, toxin and pathogen, etc. It forms the protective layer against the fluctuating environment, such as strong UV radiation. When UV reaches the inner layers of skin, it triggers the production of the enzyme collagenase, and this leads to the degradation of the collagen fibers. Therefore, skin is considered as the first line of defense of UV light from the sun. Under the absorption of UV radiation and the production of energy involved oxygen, reactive oxygen species, ROS, are present in our body. They damage cells and reduce the activities of cells. All this leads to the oxidative stress to some useful substances in our body, such as lipid, proteins, or DNA. And finally, the general aging process takes place. Luckily, we have certain level of coenzyme Q10 present in our body for combating the process. Coenzyme Q10 belongs to the group of lipid-soluble benzoquinones, and it is present in the hydrophobic interior of phospholipid bilayer of cellular membrane. It possesses various functions in human body in both medicinal field and cosmetic field, such as it helps improving the condition of the patients suffering from cardiovascular disorders, migraine, and Parkinson. When facing the problem of aging, it acts as an antioxidant and helps enhancing the resistance of the skin by removing the free radicals like ROS. German research group has proved that it can also limit the action of collagenase by suppressing it from damaging the connective tissue in skin, and resists UV-mediated oxidative stress. It contributes a lot in skin protection against early aging, wrinkle formation, and also loss of cell formation. But from the previous study, it shows that the synthesis of coenzyme Q10 by body becomes weakened after the age of 35 to 40, and this leads to the deficiency and aging problems. In the past, there is a considerable amount of research has been carried out to prove various functions of coenzyme Q10, but little research has been focused on the antioxidation of coenzyme Q10 for the women, particularly in the age group of 35 to 40, since the synthesis of coenzyme Q10 is weakened in this period. Therefore, in this project, we investigate and compare the effectiveness of antioxidation of coenzyme Q10 in 3 different age groups for women. They are 28 to 34, 35 to 40, and 41 to 46. and so as to give a general picture and to show the specific relationship between the effectiveness of antioxidation of coenzyme Q10 and women in different age range. And in this project, there are altogether 2 tests will be carried out. Since the clearest demonstration of the photoaging is the presence of deep wrinkles, so the first one is called quantitative microtopography. It is used to observe the depth of the wrinkle in the skin. 3 groups of women will be applied the cream containing 0.3% of coenzyme Q10 around the eyes daily, once daily, for 6 months. Both mean peak of the valleys of the peak of the wrinkle and the area of the peak and the troughs have been taken into account for calculation. And the second one will be the measurement of size of corneocytes. Corneocytes make up the outermost layer of the skin, and it is repaired and replaced by keratinocytes. So therefore, its area is proportional to the time taken for keratinocytes to differentiate and move towards the outermost layer of skin. This implies that the time will be longer, much longer, for the keratinocytes to move through the layers in aged skin, and the corneocytes become larger. In this experiment, for each group of women, we will compare the initial average surface area of the corneocyte and the final area after the injecting of 0.3% of coenzyme Q10 daily into forearm for 20 weeks. And here are some expected results. By using the QM quantitative microtopography, a reduction in the depth of wrinkles in skin is found, and the most obvious result may be found in the oldest group, which is 41 to 46. This may be due to the low level of coenzyme Q10 present in the body, and also benefits from the coenzyme Q10 supplementation. And for the second one, it may be found that there is a decrease in the corneocyte area over time, but the greatest percentage of change in area is found in a group of 35 to 40. This may be due to both antioxidation of coenzyme Q10 and also the metabolism in the body. And here are some references of the article. That's the end of my presentation. Thank you.